This is Group 8's Broccoli Enzyme Activity Lab.
So what is being tested? The question is, how does substrate concentration affect the rate of enzymatic activity? Well, the purpose of this experiment is to observe the effect of substrate concentration on the rate of enzymatic activity based on the number of bubbles formed. To test this, our group researched different types of experiments and then created our own lab. In this experiment, the independent variable was the amount of hydrogen peroxide in each solution. The dependent variable was the formation of bubbles, which represents the release of carbon dioxide. Finally, the controlled variables were the size and weight of the broccoli, the same type of container, amount of soap, and the amount of distilled water.
And these are the steps to the experiment. Start out by measuring and pouring the water into the cups and then label each cup with the amount of hydrogen peroxide you will put in them. Then drop all the broccoli in at the same time and squirt 2 drops of soap into each cup. Measure and put the respective amount of hydrogen peroxide into each cup and then set a timer for 5 minutes. After the reactions have completed, make sure to clean up, throw away the water, and then repeat the experiment twice.
To reiterate, our purpose of this experiment was to observe and see how enzyme activity is affected by varying substrate concentrations. This will be concluded qualitatively by the amount of bubbles and quantitatively by the total solution height measured in centimeters.
Trends. Our null hypothesis states that the addition of our substrate, which in this case is hydrogen peroxide, would cause no bubbles and no difference between any of the reactions in the cups. Our alternative hypothesis is most supported by the data, and it decides that each cup will be different from one another. The trends of this graph show that the more substrate an enzyme has to work with, the quicker and more dramatic the reaction will be. The other hypotheses shown here just further explain the trend of how the more hydrogen peroxide, the more enzyme activity.
Prediction based on our prior research and knowledge on how enzyme activity is affected. Generally, we thought an increase in hydrogen peroxide correlated to higher enzymatic activity because of the higher foam levels and rise we saw in many of the videos. Therefore, it would be shown by more bubbles in our experiment. Our hypothesis was supported in several ways by similar experiment and research done by Scientific American. Without any hydrogen peroxide, the total solution height was 5.45 centimeters in both trial 1 with 11.7 and 3 gram broccoli enzymes. The equivalent height and visuals indicate there is no presence of bubbles which would otherwise contribute to the height. Scientific American also supported this in the same condition because if there is no hydrogen peroxide present, the catalase cannot function. Significant height difference, 7.13 centimeters and 6.48, compared to the initial 5.45 centimeters, further show that more product was formed as a direct correlation with the increased substrate amount. Scientific American supports our hypothesis of this correlation because once you add more hydrogen peroxide to the solution, the reaction rate will increase as more substrate molecules can collide with the enzyme, forming more product. It is also important to note that although there is a general increase of solution height with hydrogen peroxide concentration, it wasn't a drastic change from each cup but fairly small. For example, in trial 1 with 11.7 broccoli enzyme between cup 2 and 3 with 1 teaspoon and 2 teaspoon hydrogen peroxide concentration respectively, there was only a 0.4 height difference. This data is also validated by Scientific American, stating it should have resulted in a similar amount of foam as in cup 2, which there was because there was little to no height difference, further ensuring that the data we collected from the experiment was accurate.
Several factors in the cellular environment that change the enzyme activity. Temperature, pH, enzyme, and substrate concentration are some important factors that can change how cells function and aid in chemical reactions occurring in the body. Enzymes work most efficiently at a specific range of temperature and pH and can lose or slow down its ability to lower the activation energy if not in its normal conditions. This can be seen as one common result from major environmental changes is that the enzyme starts to denature, showing that it's losing its ability to maintain homeostasis.
The purpose of this experiment we performed was to examine the impact of the amount of substrate concentration in conjunction with enzyme activity. We used 100 ml water, 11.5 grams of broccoli in each cup at different amounts of substrate concentration, 1 teaspoon, 2 teaspoon, 3 teaspoon, and 4 teaspoon. The first error that occurred during the experiment was the imprecise cutting of the broccoli sizes, which lead to different surface areas. The leaves of the broccoli occupy more space than the stems, which results in less enzymatic activity.
Moreover, here's Feng Lin with another human error.
The second error that occurred during the experiment shows that additional water droplet clinging to the sidewall of the cup would contradict with testing of the independent variable. Having additional water droplet in the cups would decrease the accuracy and precision of the experiment. Controllable drop size of the soap being squirted into the cup. The size of the soap droplet depends solely on the exertion of forces onto the bottle, and as the stirring rod was used to evenly distribute the solution, more bubbles were formed. Therefore, it would cause the recording of data to be inaccurate.
This brings us to our future extensions. One future extension we would do is that we would exchange our teaspoon that we use from a plastic to a real teaspoon. This will make the amounts of H2O2 more consistent and make our results more accurate. We saw most of the reaction occur in the stems instead of the head. To elicit a better reaction, we would want to use the stems instead of the head and the stems. Finally, the last future extension is that gloves needed to be worn. There could have been unwanted particles and different things on our hands, and that could have affected the outcome of the experiment.